Cool. So it is my great pleasure to introduce Dr. Farzaneh Najafi as our seminar speaker today. So Farzaneh is the newest addition to the Georgia Tech New York community. So she started as an assistant professor in the School of Biological Sciences this January, so just last month. Before coming to Georgia Tech, she was a scientist at the Allen Institute for Brain Science in Seattle. That's where I actually first met Farzaneh. So she started at the Allen Institute in 2019. And I remember meeting Farzaneh for the first time at the happy hour. And then we were just like doing our intro, like, hey, I'm Hannah, hey, I'm Farzaneh. And then COVID happened, so we didn't really get to interact. But I'm really glad that she is now here and we can interact more. So before she was at the Allen Institute, she did her postdoc within Churchland at Cold Spring Harbor. And before that, she did her Ph.D. at University of Pennsylvania. So with that, please welcome Farzaneh.
Can you ask people on Zoom to make sure they hear me?
Yeah, it doesn't matter.
Okay, awesome. Hi, everyone, and thank you, Hannah, for your introduction. Yeah, as Hannah mentioned, it's less than a month that I've been at Georgia Tech, the School of Biological Sciences, doing neuroscience. I'm going to present the work that I've done during my PhD and at the Allen Institute. If any of you happen to be at my job talk, half of the talk is similar, the other half I replaced by my newest stuff. And then, what is it? That's why. Is it better? Okay. I'll try to speak louder. So what I would like to do for my lab to study how prediction signals are generated, and I would like to study that for cortico-cerebellar circuitries. Our brain does not just process the sensory inputs, but it also generates predictions and updates predictions about the world. Predictions are important for our survival and adaptation in a rapidly changing environment. They influence our motion, perception, and cognition. For motion, what I mean, let me use my cursor, is that imagine you want to reach for a cup. The way you adjust your movement, your muscle force, your grip is based on your brain's prediction of what this cup is made of. You will do it differently for a cup that is made of plastic versus the one that is made of glass. So even though you may not be aware of it, your motor system is doing that while you reach for an object. Predictions affect our perception. And what I mean by that is that, for instance, here, all you see from a representational perspective is white and black patches. But what your brain, in most of you guys, I assume, sees is a soccer ball. This is because of your prior exposure to this pattern that now these predictions influence how you perceive the sensory input. So this is what I mean by predictions influencing our movements and our perception. They also influence our cognition. And for instance, if you're driving and a deer all of a sudden appears, you need to make a cognitive choice, whether to stop, whether to change direction, or slow down. So this comes from your predictions of how fast the deer is moving, how far you're from the deer, and your prior exposure to driving and such scenarios. So I hope I made it clear why predictions affect every single instance of our life. When there is an error, meaning that there is a discrepancy between the predicted and the actual input, we call it a prediction error signal, and they drive learning or updating predictions. We can call updating predictions learning. They're literally the same thing. For that example of the motor system, when you reach out for a cup, if you apply the wrong amount of force because somehow you thought this cup is not made of plastic but glass, then you'll end up squeezing it too much. So this is a prediction error. Your brain learns, and the next time when you reach for that object, you will apply the right amount of force. Or in terms of perception, imagine a kid who is not aware, or anyone who is not aware of light refraction. If you see this straw in the cup, you might think that the straw is broken. But then prediction error happens, it will be updated, and you will learn that no objects that pass through water will appear broken because of this physical phenomenon. Or in the case of driving, if you made the wrong cognitive choice because you had the wrong prediction about the distance of the deer or the speed of the deer, you'll encounter a prediction error, and that will update your priors and how you drive next time when you see such an object. So prediction errors drive learning, adaptation, or updating of predictions, and this happens constantly in our brain. Why does this matter? Because if generating and updating predictions, which we will refer to as predictive processing, if this is impaired, it leads to perceptual and movement disorders. For perceptual, a clear example is what happens under psychedelics or in psychological disorders like schizophrenia. The idea is that you are disconnected from the external world because you're making predictions that don't match the actual world. So it's like you have an internal world that has nothing to do with the external world. As a result, we will see things or hear things that don't exist, and this causes psychotic symptoms. So that's what we refer to as perceptual problems. Or when it comes to movements, if we cannot update our predictions, when we are faced to a new sensory motor environment, we will have movement problems. So that's why we care about this topic, and we want to know how predictions are generated and updated in the brain. That's the vision for my laboratory. To put it a bit in more technical and formal words, our brain has a model of the world. We call it an internal model. It makes predictions. It receives external inputs, compares them. If there is a mismatch, it updates the predictions. And the thought is that, or based on the evidence, there are many brain regions in the brain that provide these predictions. Importantly, to my research topic, these predictions are also observed in the cerebellum. So we have cerebellar and cortical regions that are providing internal models of the world. There are key questions here that we would like to answer. What are the computations and cell type specific circuit mechanisms that underlie predictive processing in each of these regions? How distinct are the internal models of the world in these regions? One classical hypothesis is that the cerebellum cares about the motor system. It only provides predictions about the motor world. However, the cortex is more about the cognitive and perceptual world. Is that actually the case? And then importantly, how are prediction signals communicated between the cerebellar and cortical regions to support predictive processing? So with that introduction, I'm going to talk about the past research that I've done, which prepares me to address these questions. I will talk about my Ph.D. research, which was about investigating teaching and predictive signals during motor learning in the cerebellum of mice. I will talk about the work I did at the Allen Institute, which was also about investigating surprise and novelty signals during visual behavior. And I will skip the work I did during my postdoc, which was about decision signals for this talk. Okay, part one, prediction and error signals are encoded in the cerebellum during motor behavior. A little bit about the cerebellum. Cerebellum is involved in motor coordination and motor adaptation. It's important for calibrating simple movements like reflexes. It's important for more complex movements, walking, singing, writing, playing piano, and so on. Cerebellum is thought to include an adaptive internal model of the world. And let's break that terminology down. The idea is that, again, there's a region in our brain that contains an internal model. It receives a copy of the motor command because this region contains a model of the motor system. After the internal model receives the copy of motor command, it predicts the sensory motor consequences of it. So it says, for instance, moving like that will lead me, land my hand on the table, and I will receive this sort of sensory input. Then there is this other region in the brain called the comparator that receives the prediction and also the actual sensory motor input. It compares them. If the two are different, it generates a prediction error signal, which will go to this part that is a plasticity side. This part will receive the prediction error signal, and it will generate updated predictions that then will go to another region called the motor controller that generates the proper motor command now. And again, a copy of that motor command will go back. So there is a loop here. Now let's put these onto brain regions. The idea is that the cerebellum contains the internal model and also the plasticity site. So that's why we refer to it as an adaptive internal model for the motor system. This is a classical view. Then we have the inferior olive in the medulla. That is the site of the comparator, and its axons, which are called climbing fibers, carry prediction error signals. This is how the cerebellar circuitry looks like. We have Purkinje cells in the cerebellar cortex. These are climbing fibers, which are the axons of inferior olive neurons. And then Purkinje cells converge onto the DCN, deep cerebellar nuclei, which acts like a motor controller here and generates the motor command. This all looks interesting and straightforward. However, there is a big controversy here. The controversy is that prediction errors are graded signals. We just know that. We don't make binary errors. Errors could be a little, small, bigger. They have different sizes. But climbing fibers that carry prediction error signals are thought to be binary. So the question is that how do we get something graded out of a binary neural code? This is the seminal work that was done by John Eccles. He made the first recordings from the cerebellar cortex. And what you see here is the superimposition of multiple recordings from Purkinje cells that shows they either don't fire, or once they fire this complex pattern of spike, which is referred to as complex spikes. And it's all or none. And it's written in his work as well. So we have this climbing fiber input to Purkinje cells that evokes these complex spikes. They're binary. They're also hypothesized to carry graded prediction error signals. How do we reconcile these two? That was the question. The approach I took was one to demonstrate error size actually matters in motor learning. It is really what I said, that different sizes of error evoke different levels of adaptation. And second, identify the code in climbing fibers, the neural code for error size. A little bit about eye-blind conditioning, which was the simple motor learning paradigm that I used. It's a very simple classical conditioning paradigm. What happens is that we have a neutral signal, an LED signal that is paired with an air puff evoking signal, like a corneal air puff. Repeated presentation of these two stimuli makes a prediction such that the mouse will predict the occurrence of the air puff and blink in advance of it. Let me show it in the actual mouse. So what you see here is a video of a mouse when the mouse has not yet learned to associate these two stimuli. What happens is that the LED is presented, the mouse does not blink, that red trace is the closure of the eyelid. And then the air puff is presented, and then the mouse blinks. So what you see here, this blink is just a reflex blink that does not require learning. It's just a reflexive behavior. Now, after many exposures, this is what we see. LED is presented, and then we see a well-timed learned blink in advance of the air puff. So the mouse is able to predict the occurrence of the air puff. So we have two types of blinks here. One is this one, which is the learned blink, and the other is this one that happens after the air puff, and that's the reflex blink. This learned blink is what's known to be cerebellar dependent. We can just get rid of the cortex and still we get this learned blink. This is a sequence of days, 1, 2, 3, 4. Starting from here, we see a little bit of this learned blink happening. So it takes about 4 days of about 100 trials per day for them to show this learned anticipatory blink. Now let's go back to our theoretical diagram and see how eye blink conditioning fits onto this circuitry. The idea is that the air puff is the actual input that goes to the inferior olive. And then the learned blink is generated by the motor controller, the deep cerebellar nuclei. This is our hypothesis now, that a prediction of the air puff is coming from the cerebellar cortex. And if this prediction does not match the actual sensory input, then this prediction error will go back to the cerebellar cortex and will generate an updated prediction and a new learned blink that matches the air puff. So you might wonder, what about the air puff is predicted here? There are different features. One is the timing of the air puff, that for instance, when the LED happens after about 300 milliseconds, the air puff is coming. The other is about the intensity of the air puff, that the LED happens in 300 milliseconds, I expect an air puff of strength medium. So we chose to work on the strength of the air puff and play with that and see if we can find the model here, if we can find evidence for the model here. So changes in air puff strength will introduce prediction error signals. And we address two questions. One, does air puff strength matter in eye blink conditioning? Because now air puff strength is a way that we induce prediction errors. And two, we wanted to know if the adaptation to air puff strength happens in single trials, because based on this model, any discrepancy will update the cerebellum and will lead to a new learned behavior that matches the actual sensory input. Number one, I showed that error size matters for updating predictions. What we see here on the x-axis is trials within a session, different blocks of trials. Here, I'm indicating the strength of the air puff. One is the baseline. Here we have increased it, decreased it, and to different amounts. And this is a summary of the learned blink magnitude on each trial. What we see is that it goes up and down and up, and the magnitude of that reflects the size of the strength of the air puff. So changing the air puff strength induces prediction error signals, and that induces appropriate amount of learning. These are two examples of these actual eyelid traces. You can see that when we move from 1 to 1.8, not only the reflex blink increases, but also the learned blink, and the other way around. When we go down, both the learn and the reflex blink decrease. So hopefully it's clear now that error size matters for updating predictions. It also matters in single trials. To show that, I made perturbations or changes in air puff strength on single trials. And then I compared the learning after that perturbation with the magnitude of the eyelid signal before the perturbation. So we're going to compare this trial plus one, meaning one immediately after perturbation, with the other ones. This is the trial before perturbation. On the y-axis, you're seeing the eyelid closure. X-axis is the time. This is when the LED happens, and 250 milliseconds, that's when the air puff happens. This is the perturbation trial. That's when we remove the air puff. What do we see is that the reflex blink is gone. However, notice that we have the learned blink, the same size as before, because the mouse did not expect the perturbation. But very interestingly, on the one trial, immediately after the perturbation, we already see a small adjustment of the magnitude of the learned blink. So one trial, after a change, the mouse adapts the size of the learned blink. This is the perturbation in the opposite direction. Now, all of a sudden, in some individual trials, I increase the magnitude of the air puff. And what we see here is that on the perturbation trial, again, the learned blink is the same. However, the size of the reflex blink has increased. And on the trial immediately following that, we see an increase in the size of the learned blink. So error size matters for updating predictions and adaptation in individual trials. Now that we showed it at the level of the behavior, we want to see what happens. What is the neural code for that? For that, I chose to do calcium imaging from Purkinje cell dendrites instead of recording directly from inferior olive neurons. This is because recording from inferior olive neurons has technical difficulties. They're very close to the spinal cord and the quality of the signal will not be good. The other reason was that we thought we are not going to find a code at the level of individual neurons. It's going to be a population code. So we decided to go with a population approach. And calcium imaging allowed us to do that. You might wonder why we did calcium imaging from Purkinje cells. This is because there is something very special about this circuitry. Individual Purkinje cells receive input from individual inferior olive neurons. Because of this one-on-one correspondence, we can use the signal in Purkinje cell dendrites as a proxy for the activity of climmin fibers. Also, when climmin fibers are active, they evoke a massive dendritic activity, a very distinguishable dendritic calcium signal in Purkinje cell dendrites. So it's a very appropriate system to use calcium imaging. This is a movie that I've made from one of my recording sessions. What you see here, this is the beautiful array of Purkinje cells. We look at them from the top, and that's how they appear. So you're seeing the raw fluorescence movie here. This is the extracted delta F over F signal that just shows how fluorescence signal changes relative to the baseline. And then these are dendrites that we have identified. So dendrites appear as these stripes because that's how they are, and we're looking from the top. These are the extracted fluorescence traces. Each line corresponds to one of these dendrites. I hope you can see these large calcium transients. Each one of these large calcium transients is reflecting the activity in one climmin fiber. The lines are when the air puffs happen. So what I did here is that we have these awake behaving mice under the 2-photon microscope, and I present air puffs of different strengths, and then we are going to look at climmin fibers. So that's how we want to know how climmin fibers encode the strength of the air puff, which is the prediction error signal. Number one, let's look at individual trials. Each point here is when we detect one of these calcium transients or when the climmin fiber is active. That's when the air puff happens. I hope you can see that there is no rate code in these neurons, unlike the typical neurons in the brain. What I mean is that it's not like the stimulus happens and we see that the rate goes up. These neurons stay at 1 hertz more or less all the time. But what we see is that the probability of activity across trials goes up. And this is summarized in the PSTH here. Now let's compare that with a strong air puff. What happens here is that the probability of activity of one climmin fiber or one Purkinje cell dendrite across trials goes up. So that's great. We see that it was kind of predictable. It does address how learning happens in blocks of trials, but remember we had single trial learning. So the question still remains, how do we get single trial learning that matches the size of the error from climmin fibers? Obviously, we thought, let's look at synchrony. Maybe this is a population code, and that's definitely what we found. Here on the y-axis, each row is the percentage of coactive dendrites. So we're looking at a synchrony code. 0 is when the air puff happens, 4 different strengths of the air puff. And as you can see, for stronger air puffs, we see a higher percentage of coactive dendrites. In other words, synchrony among climbing fibers go up when the air puff becomes stronger. This was also kind of predicted because inferior olive neurons are known to be connected through gap junctions. This is great. It allows for single trial learning that matches the error size, but we didn't stop here. And the reason is that this code will be available to these deep cerebellar nuclei neurons. Why? Because they get convergent input from many Purkinje cells. So they have access to this percentage of coactive dendrites. But we wondered if individual Purkinje cells also have access to the error size in individual trials. So we were not hopeful to find the code because of this classical work that John Eccles did and suggested that almost invariably the response had an all or nothing characteristic. But that was done by using electrophysiological methods and in this soma of Purkinje cells. Remember, I'm using a different method. I'm imaging calcium signal in the dendrites of Purkinje cells. To our surprise, when I compared the magnitude of the calcium signal between spontaneous and between air puff evoked conditions, I saw that in a lot of the dendrites, we do see a graded non-binary signal such that climbing fibers actually had a larger dendritic calcium signal when they were evoked by a stimulus compared to when they fired spontaneously. Then we wondered if that is true for different strengths of the air puff. And we saw that as we increase the air puff strength, indeed, the dendritic calcium signal in Purkinje cells, the magnitude of it goes up. And this is true for all of the dendrites that we quantified. So this suggested that the activity of climbing fibers evokes a non-binary dendritic calcium signal in Purkinje cells. And especially because calcium has a lot to do with plasticity and learning, we thought that this is a way that can result in graded motor learning. A little bit about the, okay, before I move to the second part, are the conclusions from this first part of my PhD clear? That climbing fibers have a non-binary code at the level of the Purkinje cell dendritic calcium signals, which can lead to graded motor learning. Okay, so the second part, this was a collaboration that I did at Princeton, and now we were curious about this other input to Purkinje cells. I talked about climbing fibers. Purkinje cells have a second input from granule cells. Those are called parallel fibers. And then we were wondering what information they carry. So we imaged them during eye blind conditioning, and we found that the activity of granule cells very much reflects the learned blink. So this is the behavior and this is the learned blink that we've seen many times so far. And when we look at the calcium signal in granule cells, it's pretty much the same thing. It could be happening before the learned blink, or it could be just a proprioceptive type of input that leads to this signal. For that, we looked at the timing of the signal. And what we see is that in the vast majority of granule cells, the calcium signal in these granule cells actually does precede the occurrence of the learned blink. So I'm going through this, there is a lot of information in that paper, but that's the gist of it, that granule cells have this information about the learned blink, and it's a predictive signal. And we called it a feedback signal because they have to get this information from somewhere. And the hypothesis that we would like to pursue is that these deep cerebellar nuclear neurons send a copy of their of that learned blink back to the cerebellum. And this is exactly what we had in our model here. Go ahead.
I was wondering if you could just clarify where the granule cells are in the circuit compared to the Purkinje cells and the climbing fibers.
Yeah, sure, thank you. This is actually cerebellar circuitry is really gorgeous. So Purkinje cells are in the cerebellar cortex. We have molecular layer interneurons, then we have Purkinje cells, and we have the granule cell layer. So this, I have drawn it pretty much like that's about how you see, like we have granule cells and they send their axons up to Purkinje cell dendrites.
Okay, and so the granule cells and the climbing fibers are both synapsing onto Purkinje cells.
Exactly. Yeah. Thanks for asking. If you're not from the cerebellum background, the circuitry won't be clear. Yeah. Okay. So back to our model. I talked about this part. That was the first part of my PhD work. And then the second part, we're talking about this feedback signal that carries a copy of a motor command. And if you think about online motor control, for instance, when I'm reaching for something and then some perturbation happens and immediately online I control for it, this copy of motor command can very much help with that online motor adaptation. Okay, the summary, climbing fibers are not all or none. They allow for graded encoding of teaching signals. Teaching signals are also prediction error signals. They're used interchangeably in single trials. And then we have granule cells that carry a predictive feedback signal during motor learning. So after my PhD, I decided to move outside the cerebellum field because I wanted to broaden my view and not stay. Do you have a question?
Yeah, before you move on to the part two, I was just a little unclear about where you explained the probability code. How is that different from rate code? From rate code, because you said it's not rate code, but it's like the probability increases, right? But then if the probability of this spikes increases, then like how is it different from the rate code?
Right. Yeah. Rate code, I think of it as single trials, right? That in one trial, let's say the air puff is weak, you get 10 hertz, it becomes strong, you get 20 hertz activity. But here we have to sum it up across trials. So we will not have access to that in one, because here, whether the air puff is weak or strong, we stay at 1 hertz. But it's just that whether it happens or not, the probability changes across trials. Yeah, thanks for asking that. Yeah, so I decided to explore the cortical regions too, and it appeared to be very helpful because at the end of the day, I found connections with what I had seen in my PhD, and that's why now I want to study the interaction of the two regions. So at the Allen Institute, they have this large data set which is related to novelty. A little bit of information, like a historical background on novelty. This German psychiatrist von Restroff demonstrated that when humans are exposed to a novel object, they can remember it much better. So there is something about novelty that increases memory and attention. And then many studies after that, they looked at the brain and they showed that when humans see a novel object, the overall excitability in the brain goes up. We're, of course, systems neuroscientists, and we would like to know what circuitry induces the novelty effects. One good candidate is the disinhibitory circuitry that is formed because of VIP inhibitory neurons that connect to SST inhibitory neurons, which then inhibit pyramidal cells. So we have this double negative sign here. As a result, when VIP neurons are active, even though they're inhibitory, they lead to the disinhibition or activation of excitatory cells. We were wondering if this circuitry can underlie the effects of novelty. The reason we thought that could be a good candidate is that VIP neurons receive different types of input. They receive excitatory input from other brain regions. They also receive neuromodulatory input. Importantly, multiple studies have shown that when movements happen, we see that the activity of VIP neurons go up, the activity of SST goes down, and the activity of pyramidal cells go up. And this has been thought to explain the effects of movement on improving the animal's perception and the arousal-related effects of movement. So we thought the same circuitry might be involved for novelty. The question is that what is the cell type specific mechanism that underlies the impacts of novelty on the brain? And the approach we took was that we imaged the activity of excitatory, VIP, and SST neurons in the visual cortex during a visual task that involved novelty signals. This work was done at the Allen Institute. Allen Institute is great. It's well known for its open science and team science. And this paper is going to be on bioRxiv in a week or so. There are many authors on it, and among the first co-star authors are me and my main collaborators, Marina, Alex, and Irina. So this is very much of a real team work that I'm presenting here. A little bit about the specific task that we train the animals on. The images are presented repeatedly, and then at a random point, they change to a different image. For example, it's tree, tree, tree. Now it becomes flower, flower, flower. The animal is trained to detect the image change and lick to a water spout. This is how this looks in reality. You can see the image on here, and you can see that once it changes, here it changes, and then the animal licks. So I'm going to leave this on, and you can see it for a bit. Image change happens now, and we see the animal licking. In addition to image changes that are a sort of unexpected signal, there is another type of unexpected signal in this task, and that's random omissions of the images. You can see one of them upcoming right now, that all of a sudden an image is omitted. So we have image changes and we have random omissions. They allow us to study how prediction violations are coded. And then we have the novelty thing. What is the novelty? In each image, might see in each session, might see 8 images. And that's, for example, those 8 images. This happens during learning, and then all of a sudden we introduce a session where might see another set of 8 images. So it's the same task. They have to do the same thing. They take the image changes, but now they're seeing other images. So it's an entirely new context. Also, the stimuli are new. So we call these sessions, this is the familiar session, this is the first novel session, and this is the novel session that came after that. And it allows us to see how transiently the effects of novelty are. That's the behavior. I hope the components of the behavior, image changes, omissions, and the novelty are clear. And this is calcium imaging from 3 distinct mouse lines. Please don't get confused. Here, we have not imaged these 3 cell types in the same mouse. That would have been awesome, but this is not what I'm going to present. These are 3 distinct mouse lines, and then we are just putting our hypotheses and interpretations together. So let's look at how these 3 cell types respond to image changes in familiar and novel sessions. This is what VIP neurons do. In the familiar session, they really don't respond to images. This is the image, and this is the image change. A little bit more increased response to the image change, but not much. They're not much image responsive in general, VIP neurons. Now it's a novel session, and all of a sudden they become very image responsive. So VIP activity changes from non-image responsive to image responsive in the novel session. SST neurons are in the familiar sessions very much image driven, and the opposite happens. They become very small. They have very small activity in the familiar session. And remember this inhibition here. How about excitatory neuron? Now their activity is closer to VIP, meaning that they become more active in the novel session. Remember, this is what I showed in the very beginning to show the previous literature, that the brain in general, the activity goes up in response to novelty, and that's what we see in excitatory neurons here as well. Yeah.
I was wondering if the excitatory neurons are in a specific layer, or whether, like,
Great
whether this is
question. Yes.
represented.
So this is all in the 20 supplemental figure that we're going to have. Yes, we have recordings from 4 cortical depths and 2 visual areas, V1 and LM. I'm showing everything here. There are some layer and area differences. I do have a hidden slide that I can get to, but it's limited. If I want to summarize it, novelty effects are more seen in LM, which is a higher order visual area, and superficial layers of that. In this talk, I'm not going to get to area and layer differences. Yeah, but whatever I'm showing we've observed it for all layers and areas. It's just the magnitude of that effect is different a little bit. Okay, so image responses are increased in VIP and excitatory neurons in novel sessions. How about omissions? When we look at VIP neurons, we see that in the familiar session, again, not much image response, but a very large omission response. So it's like these VIP neurons are kind of omission detectors or surprise detectors. Something very unexpected happens and they fire. But what is really, really interesting is that when we go to the novel session, now they don't care much about this image omission anymore. Instead, they just care about the images that happen. This has been a very interesting finding. I have had a couple interns that we tried to figure out what this is about. And this is one direction that I will pursue in my lab to figure out how these novelty effects on VIP neurons happen and why do we turn from an image responsive mode to an omission responsive mode. In SST neurons, in neither familiar or novel session, really, we see an encoding of omissions. And in excitatory neurons, just sort of like that disinhibitory circuitry, we see that in the familiar session, they do encode these surprise omission signals. So as I said, I would like to look at these omission signals as a prediction error signal. The mouse expects to see an image every 750 milliseconds. Now, all of a sudden, there is no image. So there's a prediction error. If it is the classical predictive coding hypothesis, then we expect this prediction error signal to be stimulus specific, meaning that was it image 1 that was omitted or image 2 that was omitted? We expect to see a difference. So what I did for that was that I trained a classifier to see if the omission signal in VIP neurons is different, whether the omission happened in between image 1 repeated presentations or other images. What I saw was that the gray is the shuffle control, and this is familiar, this is novel. I did not see any image specificity in the omission response of VIP neurons. So what this suggests is that it could be a broad, non-specific prediction error, non-image specific prediction error signal, or it could be a temporal violation signal. It could be neuromodulatory driven. And these are the directions that I would like to pursue in my lab. In SST and excitatory neurons as well, we did not see much of specificity to this surprise omission response. Okay, so let's go through an interim summary. At the population level, and I'm focusing on that because the rest of the talk is going to be on single neurons, we see that novelty changes the activity of excitatory and inhibitory response neurons. But what we want to know is that how do individual neurons, how does their activity change with novelty? I have just pulled everything together. For that, we took advantage of this feature in Allen Institute data that we had tracked the activity of individual neurons across multiple sessions. So we can just go take one neuron and see how this neuron is active in the familiar session and how it is active in the novel session. Our team did two types of analysis, one generalized linear models to see what are the features that drive the activity of individual neurons. And then we use the output of this model to cluster neurons and see if we have different functional subtypes of neurons based on how their activity is modified with novelty. The generalized linear model was defined the activity of a neuron based on images, omissions, behavioral features like pupil diameter, running, licking, and task features. So let's look at these four categories of features and see how individual cell types in the visual cortex encode them. VIP neurons encode images. SST neurons have a strong encoding of images, also excitatory neurons. For omission, it's only VIP neurons. We don't see much in SST or excitatory. For behavior, VIP again has a significant encoding, but not excitatory or SST neurons. So images are most strongly encoded in excitatory and SST neurons. Omission and behavioral parameters are most strongly encoded in VIP, and task parameters I didn't talk about. It's pretty much weak everywhere except a little bit for excitatory neurons. In this slide, I'm not talking about novelty. This is just the general coding properties of these neurons. And now I'm going to talk about novelty. As I mentioned, the activity of one neuron tracked across familiar, novel, and the subsequent novel session. And then we use that to define the GLM, and then we use the output of the GLM to cluster neurons. This is a complex slide. Bear with me. I'm going to walk you through it one by one. So we have 4 types of features in our GLM model, right? Image, behavior, omission, task. And then we have 3 sessions: familiar, the first novel session, and the subsequent novel session. So it's a 3 by 4, it's a 12 element vector. And then each row here is 1 cell. So what you're seeing is the output of the GLM, basically the coding score, or how much the neuron is active for each of these features and each of the sessions. Let's look at this one. This is the output of the clustering. I'm not getting into the details of the methodology here. After we clustered, we found this large cluster of VIP neurons, cluster number 1, that responds more to the novel session, to the images in the novel session, and it doesn't do anything for omission, behavior, or task. We also found these 2 clusters that have a little bit increased activity to the novel session, and they also respond to omissions in the familiar and the subsequent, the other familiar session. So I hope you can see here that that picture that we got from the population averages, which seemed like all VIP neurons increased their activity to novel images and increase their activity to omissions in the familiar sessions. They're actually, when we look at individual neurons, they seem to be largely 2 subsets of neurons: the ones that increase their activity to novel images and those that increase their activity to omissions in the familiar sessions. And there is a lot more complexity, especially for VIP neurons when we look at the other clusters. Like this is a behavioral cluster of neurons. We have this very interesting cluster here that actually responds to images more in the familiar session, completely opposite to what I showed you in that simple average figure. So I hope this figure shows you that there is a lot more complexity in the activity of individual neurons when we go through this sophisticated analysis. For SST neurons also, we have these clusters that, just like the largest cluster, just like the average, responds mostly to familiar images. SST neurons don't do really much about any of the other features. But again, I want to focus your attention on this other cluster here of SST neurons that actually likes novel images more, unlike what we saw in that simplified average picture. Excitatory neurons, we have this large cluster that loves the novel images, but also notice this large cluster that we call it the null cluster. This cluster does nothing. So we have all these excitatory neurons that were not evoked by any of these features. And then see this other cluster that actually likes the familiar images more. So what I want to show you is that at the Allen Institute, they're very much interested about cell types. They have transcriptomics data, morphology, and electrophysiological data. And now our team showed that when we look at the coding properties of neurons at the functional level also, there are all these functional cell types. And the next project at the Allen Institute, which unfortunately I won't be there, is that they're going to bring these 2 together. We have functional cell types, we have MET, the morphology, electrophysiology, transcriptomic cell types, and then they want to register those 2 to see if these functional subtypes of, let's say, VIP neurons that we saw, do they have different transcriptomics? Do they have different morphologies or electrophysiological properties? Okay, summary. Novelty effects on image coding might involve disinhibition, at least at the level of the population, we see that. However, individual neurons, they're much more complex than that. It could be because they have distinct inputs, or it could be because they have different genetic profiles. And then when we look at VIP neurons, they're very interesting, and they might actually, through this disinhibitory circuitry, gate plasticity in excitatory neurons when there is some novelty. So what do I want to do in my lab? I want to come back to this circuitry. The reason is that I saw prediction signals in the cerebellum, also in the visual cortex, and I want to bring these 2 together. So the idea is that this motor controller could be the motor cortex that receives the predictions from the cerebellum, that is like the forward model, and then it generates updated motor commands, sends them back to the cerebellum, and this happens. So I want to test this hypothesis. cerebellum and motor cortex, or the cortex, are connected through the midbrain, through pons and medulla, and then cerebellum is connected to the cortex via the thalamus. So there are multiple anatomical pathways that connect these two regions. And the key questions are at the circuit and information coding, are about the interaction of the two regions, and also about the causality. Is it that prediction signals are generated locally in one of these regions, or do we need input from the other region? Or could it depend on the modality of the prediction signal? I mentioned we have at the level of the movements, perception, and cognition. If we consider all of these three different types of behavior, how does the interaction of the cortex and the cerebellum vary? So these are the type of questions that I'm hoping I can pursue. I will be using closed loop visual motor behaviors, and I will be imaging the activity of specific cell types in these regions, and I will use optogenetics and tracing to study causality, and computational methods, of course. So acknowledgements. My PhD was done at University of Pennsylvania, also collaborations at Princeton University. My PhD advisor, Javier Medina, he's still like a great mentor for me, and my great collaborator, Andrea Giovannucci and someone at Princeton. At Allen Institute, the main people I interacted with Rashawn Olsen, Marina Garrett, Alex and Irina and Jerome, as well as also Stefan and Anton and Christoph have been great mentors there. And I didn't present my work, my postdoc work, which was at Cold Spring Harbor, and collaborations with Columbia and University College London. Okay, thank you for your attention. I guess we are 10 minutes after sunset.
There's a question on Zoom from Sina Dadbiri about the first part of your talk. I'll let him unmute himself.
Hi, thank you so much for the nice talk. I had a question for the first part of your talk. And I was wondering about the olive cells and what would happen if we lesion the olive cells. Do we get rid of the eye blink conditioning?
If we lesion the olive, did you mention? Yeah, yes. So if we lesion the olive, especially during learning, yes, eye blink conditioning will be gone. After learning, no, because already prediction error signals are used to induce learning. That's a great question. Yeah, olive will be important during learning.
Okay, great. And then how about the granule cells? Like you mentioned, they're involved in motor learning feedback. If we lesion them, would we get rid of the motor learning
So
feedback?
the classical hypothesis for granule cells is that they carry the information about the LED, that conditioned stimulus. So we very much need them during learning, and also after learning, we need them for carrying the CS signal. And I'm sorry, I should say this. Also, after learning, we do need the inferior olive for carrying the air puff, but it's not like that that error puff change, like the prediction updating, we don't need it there. Is it clear? I guess I confused you. So we need both of them for the CS, the LED, and the air puff signals. But once the learning happens, we don't need them for any prediction updating, unless we are again updating the inputs.
Got it. And then how about the role in disease, like during, like for example, Parkinson's? Because the learning has already been done, would we see some, or that would be more the interaction? Yeah.
Yeah, I actually don't know. I mean, Garrett's announcement at the beginning made me also think about Parkinson's, but yeah, I don't know yet.
Yeah. Okay. I mean, it's very interesting because in Parkinson's field, they're also looking at cerebellum more and how it interacts with the like subthalamic nucleus, and it would be interesting to see the relationship.
Yeah. Thank you.
Thank you.
Yeah, go ahead.
I have a question. So first of all, thanks very much. That was great. So I have a question about the first part of your talk. You made a clear distinction between the binary sort of nature of the perceived climbing fiber responses versus the fact that error signals need to be graded in some way. But for something like an eye blink, it is kind of binary, right? It either exists or doesn't. But I definitely appreciate when you introduce the strength of the eye blink as as something that is graded. So maybe what I'm curious about is we usually think of errors as and learning is involving some kind of as an outcome that is somehow linked to the task. So if you do a task incorrectly, you don't get a reward. There's the error is linked to some kind of consequence. What's the consequence of the animal mispredicting the magnitude of the puff?
Great questions. First of all, it is totally true. A lot of people do think of blanks as binary, and that was the first part that I had to show, that actually the size of the blanks are not binary. They reflect the magnitude of the air puff, and I hope the data was conclusive that it's not binary. About why it is important you can imagine if the air puff is weak, why would the mouse do a very strong blank that might make him susceptible to other dangers, let's say in the environment? So I do see evolutionary benefits to that when it is for something that is a small danger, you don't want to completely close your eyes. Yeah.
And also maybe relatedly, do you think of if you were trying to predict the time of an event, let's say of a puff, and it was randomized, time is continuous, right? So but it's also a discrete event. So how do you like to think about
there was another student in the lab that was pursuing the timing line of these things, that yes, very much there also the timing of this learn blank reflects the timing of the prediction of when the air puff comes. Yeah. Yes. So it's not like they start closing their eyes anticipating an air puff. They close it a bit beforehand. Yeah. Go ahead.
I had a question about the graded responses. So you mentioned that the ascending fibers are 1-to-1 with Purkinje cells.
Yes.
But then you said that the grading happens by like aggregate across the ascending fibers. I was just wondering how the graded response happens in the Purkinje cells.
Okay, great question. That was exactly the point, that this part here, remember, this slide, this is about the individual Purkinje cells. And that's what I showed, that even though historically they are known to be all or none, in individual Purkinje cells, when we examine the dendritic calcium signal, that was not all or none. And that's this slide.
Okay, yeah, but how would that happen if they're only being
okay, great question. Yeah, so many studies now have come up. They show that the spikelets in inferior olive neurons, like there are these complex spikes also in inferior olive neurons, the width of those spikelets and the number of spikelets vary given the strength of the air puff. Yeah, and that's what supposedly drives these different dendritic calcium signal magnitudes. Yeah.
Okay, thank you.
Sure.
I had a question about the types of visual stimuli that you used in the second part of your talk, and like what the statistics of the images were. Like you talked about this GLM you did where you said like, oh, okay, different cells split into different categories, but
yes,
if it's say just grading stimuli of different orientations that are familiar and novel, you can see how those might fit into some of those different categories. So I was wondering if you could talk about the statistics of that.
Yeah, we have natural images here, and it's not easy to talk about their statistical features. It's not like we have changed one feature from one to the other. In the supplemental figures of this paper, also a couple papers that Allen Institute published beforehand, they've gone through, like, instead of calling just an image change, through all these possibilities, like 8x8 matrix, that what changes to what. So there is some difference, like whether we go from like this image to this image, which of course they look similar even for us as humans compared to this image to that image. Of course, there is some change. What I want to say is that our overall results about the novelty, omissions, and all of that, that was not really, that doesn't depend on what image and what features we change to the other feature. But the behavior of the animal is definitely dependent on that, and how much, like the two, they call it image similarity matrix. It's not, they have tried to quantify those. I don't remember off the top of my head. That was your question.
Okay.
Hi. So I had a question about visual processing. My understanding is that it can either be a passive process or a very active and therefore more predictive process. I was curious in the case of passive processing, because I imagine for these mice, it's a relatively active process. Their attention is probably very focused in this task. If you would expect there to be a general decrease in response to novelty because there's less visual attention, or if you would expect there to be an impact on a specific cell type in that circuit.
Thank you so much for asking that question. That's a great question. In data from Allen Institute that is also public and anyone can use, we do have passive sessions there. So active sessions are the ones that emit changes. The animal is water restricted, motivated, and they lick once the image changes. Passive sessions, we do the exact same thing, except we satiate the mouse beforehand. We put them on the same behavioral setup. They see the images, and there's no water spout in front of them. So exactly like what you're asking. We decided not to put that data in this paper, but I spent quite some time on analyzing that data. Yes, we do see novelty effects also in the passive session. So even though there is no motivation for them to attend to these image changes, pretty much all of this, we see it there too. So that's why I think a lot of these are, especially that the task is simple. It's not like we are asking them to, like to what Ming-Pai was asking, distinguish which image change to what. It's just an overall image change. So given the requirements of the task and all of that, because it's very basic and simple, we see the novelty effects also in our passive sessions. Yeah.